Okay, this is part 2, studying life. So how do we go through the scientific process? So scientists use a process of inquiry that includes making observations. So we can make all kinds of observations about the natural world, and usually this leads us to asking a question. And so we ask a question, why does this happen? What is the cause of this? Is this related to this? And so then we form a logical, testable explanation that we call a hypothesis. And then we test this hypothesis. So in testing a hypothesis, more observations may inspire the original hypothesis or maybe even forming a new one. And so let me go back for a second. The Campbell biology textbook doesn't give the standard like scientific method. It kind of explains this as just like a process of these basically 3 steps. But I am going to lay out, the typical scientific method. So step 1, similarly, making an observation. You make an observation about something that happens. This leads you to ask a question. Why does this happen? What causes this? And then you form a hypothesis. So again, the basic same steps. You form a hypothesis that attempts to answer that question, and it can be right, it can be not supported. And so this hypothesis, you may end up coming up with a prediction. So if this hypothesis is true and this causes this, if I were to test this, then this would happen. So you're making a prediction, and again, it might be supported, it might not be supported. You do an experiment to test that hypothesis and prediction. So you do an experiment, and then you look at the results. You analyze the results, and whatever data you collect can either support your hypothesis or not support your hypothesis. And that's the reality, honestly, that it might not be supported. And either way, you analyze the results and then you report your results. And then that might lead you to a different thing. So maybe you have to revise, and you have to revise your hypothesis, or maybe you revise your experiment, and then so you try again. So just to kind of touch more on that. So biology begins with careful observations. Observations can reveal valuable information about the natural world. And sometimes that observation will come from ourselves, but sometimes we'll also include observations from others. So biologists also rely heavily on what we call the scientific literature. So it's not just like literally books about science. It's science that is reported for other scientists, the published contributions of fellow scientists. And so by doing this, they build on the foundation of existing knowledge. I'm going to touch back on this because it is a cycle. So biologists will also do research on what other scientists have observed and tested, and that might lead you to your own question. So when you're testing a hypothesis, you will do an experiment and record observations, and so we call those recorded observations data. And there's different kinds of data that you can record. So qualitative data describes like a quality about something. They're often recorded descriptions. Versus quantitative data. Quantitative data describes numbers, so numerical measurements, such as the length of something, how many times something happened, the duration of something, the mass or the change in mass. So something that has to do with numbers, quantitative data. And we tend to show and organize these observations into tables and graphs. And so they do kind of start out the same, QUA, but to help you remember which is which. Especially if you speak Spanish, you can kind of rely on the Latin word origin. So qualitative, kind of similar to like if you were to describe something, if someone was like, oh, what are you looking at? Like you describe an aspect of that thing. Versus quantitative data, that's like cuánto, like how much, how much does it weigh, for how long, there's some kind of quantity. So those are two major kinds of data that you can collect. And so, for example, here's this scientist, Jane Goodall. Rest in peace. She recently passed away, but so she was, she studied the behavior of chimpanzees. And so she made observations about chimpanzee behaviors. And some of this was qualitative. You know, she described, she was able to name and get to know the individual chimpanzees. And so she described their behaviors, you know, the name of this one and interacting with this other one. And this is what they do. She's describing things, right? So that would be qualitative data. But she could also collect quantitative data, like maybe weigh them periodically or test how long they were in a social interaction for. So there's different kinds of data you can collect. And so when you collect all this data, scientists can analyze data using statistics, and this helps test whether these results are significant, statistically significant, or if they're due to random chance. So in science, the term significant means something different, not just like big or interesting or important. If something is statistically significant, that means you've run specific tests and it proves, or maybe not 100% proves, but it's most likely, more than 95% likely, that this is not due to random chance. This is some kind of relationship between this thing and this other thing. So statistics help show those things. And in science, a hypothesis, again, is an explanation based on observations and assumptions that leads to a testable prediction. And it must lead to predictions that can be tested. You can't say, for example, I think the textbook says there is a light that you have on your desk, and this desk lamp isn't turning on. Maybe ghosts mess with it. Maybe it's haunted. That's something that you can't really test. And so that's a distinction. You know, a hypothesis and prediction have to be testable. So an experiment is a scientific test carried out under controlled conditions. So it's not necessarily like Jane Goodall. She did a lot of observations, but an experiment is where you're controlling the conditions. And we actually use the scientific method and the scientific process often, especially when things go wrong. So in everyday problems, we tend to actually go step by step. We make some kind of observation about something. This leads us to a question like, why isn't this working? What is that? And so here is this example. So the observation that your desk lamp doesn't work for some reason. So that leads to the question, why doesn't it work? And you can have more than one hypothesis. You know, there's more than one thing that it can be due to. And so one hypothesis could be that the bulb is burnt out. So maybe you've had this lamp for a long time and now it's not working. So maybe the bulb is burnt out. Maybe if you actually just got this lamp and it doesn't make sense that it would be burnt out already, you would have a different hypothesis. So maybe the bulb is not screwed in right. I just set it up yesterday. Maybe I just didn't set it up right. So then you can test each hypothesis. So if you go with hypothesis 1 and you're thinking the bulb is burnt out, then if you can make a prediction. So if my prediction is correct and the bulb is burnt out, then replacing the bulb will fix the problem. And so you test a prediction. If you were to replace the bulb and then the lamp works, then your hypothesis is supported. This one is. Say that didn't work, and so you try out the other hypothesis. So the bulb is improperly screwed in. Then if that's true, if the bulb is improperly screwed in, then when I reinstall the bulb, it'll fix the problem. And say you do reinstall the same bulb, but it doesn't work, then that hypothesis is not supported. So sometimes that's kind of what I mean by you might have to change your hypothesis. If you try out one and then it doesn't work, okay, let me revise my hypothesis and test that out. And again, a hypothesis must be testable. So a hypothesis that ghosts mess with the desk lamp can't really be tested. And so when we talk about science, we only talk about natural, testable explanations for natural phenomena. There are other, hypotheses or other like realms of study as well that are, valuable, but not in the realm of science. So, for example, supernatural and religious explanations aren't really out in the bounds of science. They're outside the bounds of science. They're not really testable. And so we can't really consider those kinds of things in science. So when we typically teach the scientific method, it's like idealized. It was really like linear, perfect step by step, but it's not always like that. It's very fluid, actually. So very few scientific inquiries actually like rigidly adhere to this approach. So this figure is what the Campbell biology textbook kind of suggests, is that it's more cyclical. So exploration and discovery is kind of the same as like observations. And that might lead to forming and testing hypotheses. But then in doing that, you might have different kinds of things interact with these hypotheses and make you revise your hypothesis or revise your experiment. Maybe your experiment didn't test something as well as you thought it did. There were variables that were like outside of what you thought. And so you have to revise your experiment. So even between the forming a hypothesis and testing a hypothesis in the middle circle, there's 2 divisions. It's a whole process, and you're constantly going to get feedback from maybe your community, like other scientists, or if you are doing research, you're working with other, maybe you're working with other undergraduates. If you're doing undergraduate research, you might be working with graduate students, and then you'll also be working with a professor. So you're going to get feedback from them as well, and then that might lead you to reform this or revise your experiment. Or you might have some kind of idea. So it's just not as linear, basically, is what it's trying to say. But this is the scientific process, and it really is like that. So, for example, when I did my master's research, or even when I did the undergraduate research, there were groups before me and students before me that did some kind of experiment. They formed and tested a hypothesis, and then they analyzed their results, and maybe that led to more questions in the next project. And so then if you're doing research, you'll usually pick up somebody else's project based on what they found or what questions they ended up discovering after their experiment. And so it's a whole cycle. It really is a process. And so here's an example of a real life study done. And so this is what we call a case study when you kind of focus on one real life example. So this is a case study on scientific inquiry. And so what this group did is notice that mice will be different colors. Even if they're the same species, they'll be different colors. And they tend to be similar to the environments that they live in. So color patterns of animals can vary widely in nature and even between members of the same species. So there's 2 populations of mice of the same species. So again, this is the scientific name. The genus is the first word, and then the species is the second word. So this is Paramyscus poliognathus. And so these are the same species, even though they look very different. They have different color patterns, but they're the same species. And they live in different environments. So this population of the species lives in a beach area. So if this is Florida, it's here closer to the beach. And this other population that's more inland is more brown because they live in a brown environment, whereas the beach population are very light tan because they blend in with the white sand. So they have different color patterns and reside in different environments. The beach mouse lives on white sand dunes with sparse vegetation, so there's not a lot of stuff to hide behind. And then the inland mouse lives on darker soil. And so the two types of mice match the coloration of their habitats. And the natural predators of these mice are all visual hunters. So they focus especially on sight and seeing their prey. So this group, their hypothesis was that these color patterns evolved as adaptations that protect the mice from predators. So their prediction was that mice that didn't match their habitat would be preyed on more heavily than mice that did match the surroundings, which kind of makes sense, right? So they tested this prediction. In this case, they built models of mice. So instead of just, putting mice in different areas, and especially you don't know how they're going to behave, they built models of these mice and they painted them, tan or like a little darker brown. They painted them to match one of the surroundings and then placed equal numbers of each type of model in each habitat. And then they recorded the signs of predation. So how many times a predator tried to collect, this mouse model. And so what they found was that the data actually fit that key prediction of that camouflage hypothesis. So in the next slide, we see their actual results. So on the left panel, we're focusing on the mouse models in the beach habitat. And so there were the light-colored models that were camouflaged. And then also in the beach habitat are the non-camouflaged dark models. And so they wanted to see in this beach habitat which one would get attacked more. And what they found was that the light models got attacked less on this light beach habitat. And then the dark models did get attacked more on this beach habitat. And then on the right-hand panel, they're comparing the light models and dark models in the inland habitat. So the light models in this case on the inland habitat that's darker soil, they were predated on more and attacked more than the darker models that kind of blended in with their surroundings. So now we can apply some more terms. So in a controlled experiment, an experimental group is compared with a control group. So a controlled experiment, you have at least two groups, a control group and a experimental group. And there might be more than one in each. So the control group, you kind of have as much like normal as possible. And then the experimental group, you're changing one thing and then looking at the effect of that change. And you can have more than one experimental group. You might even have more than one control group. It depends on your experiment. And so the experimental variables are things that you can change in an experiment or will change as a result of what you change. So experimental variables are features or quantities that can vary in an experiment. The independent variable is the thing that you change. It's the one that is manipulated by the researchers. And then the dependent variable is the one predicted to be affected in response. So it's the thing that changes after you change something. So in this example, the control group is the mouse model that is colored the usual color for that environment. So the camouflage models, like for example, the light colored mouse in the light color beach habitat, and the dark model mouse that's in the inland habitat, because those are in their kind of natural habitats, those are the control groups, and you want to compare a difference, a change. And so the experimental groups, in this case, are the mouse models that are in a different environment. So the non-camouflaged mice models, like the light-colored model in the inland habitat or the dark-colored model on the beach habitat. Those would be the experimental groups because you changed something. It's the coloring in the environment. And in this case, the dependent variable would be the percentage of attacked models. So the independent variable, the thing that you changed, was the color of the mouse in an environment. And the thing that changed as a result, the dependent variable that depends on what you changed, is the percentage of attacked models. So in general, another term that is different than what we typically use this word in daily conversation is theory. So in science, a theory, in conversation, we use theory as like a prediction or a hypothesis. But in science, it means something different. A theory is broader in scope than a hypothesis. It is general enough to lead to many new testable hypotheses, and it's supported by a large body of evidence in comparison to a hypothesis. So yeah, in daily life, we say, oh yeah, I have a theory that this, but it's kind of closer to what we mean by hypothesis. So in science, a theory is supported by a large body of evidence, and yeah. So for example, the theory of gravity, it's not just like, oh, it's a theory. In science, a theory is pretty widely supported. And, scientists are, or should be, open to being wrong. Scientists will sometimes modify or even reject a previously supported theory if new research consistently produces results that don't fit it. So, for example, there used to only be two kingdoms, eukarya and prokarya, but now we know that there's three domains. There's bacteria, archaea, and eukarya. So eukarya is still a big group, but we learned that there are differences between bacteria and archaea, and they're big enough differences that they should be two different groups. And so we refined this theory. The same for, like I mentioned, plants and fungi. They saw fungi and mushrooms growing in an environment close to plants, and they thought, oh, maybe it's a plant. But no, with more information, they refined that, and now it's its own group. Plants and fungi are separate. And as I mentioned, most scientists work in teams or groups. Usually there will be graduate students and then undergraduate students. So undergraduate students include anybody that hasn't yet, graduated with a college degree. Graduate students typically have graduated from one at least, and then are pursuing another degree. So there will usually be a mix of undergraduate students, graduate students, and then the professor. They call the PI. It stands for principal investigator. That's the professor. That's the head of like a lab or a project. Sometimes some experiments will include more than one PI. So yeah, even though we kind of I always thought that scientists kind of work in a lab alone. Most scientists do work in groups. And so good communication is important to share results. And so scientists share results through seminars, publications, and websites to help say, hey, like this is what I am interested in studying. This is the question that I had. This is the research I did, and this is what I found. And they can learn from each other and help support each other by communicating these results. And so when you do this, when you publish data in scientific journals, these research papers are not published until they're vetted by colleagues in what's called the peer review process. And so if you take a class, you might have done like peer review, but it's not like that where it's just like, oh, I suggest a period here, or oh, great job. You're kind of just like saying whatever. In science, the peer review process is very rigorous. They won't just look at grammar and stuff like that. They'll look at like, how is your scientific design? What is it that you're trying to solve? Hey, hasn't this been done before? Like, what are you doing that's different? Maybe rewrite this whole thing. Maybe you should add a whole nother control group because what you did is not that convincing. It's very strict, basically, and rigorous. And usually a paper is peer reviewed by multiple peers, not just one. So it's a pretty high standard to have something that's published and is peer reviewed. And so you can publish data like that through scientific journals, and that's one way. So here's an example of a scientific journal called Nature. That's a huge one. And so different research groups will try to publish their research in here. And there are different scientific articles and journals that are more specialized. So this includes anything in nature. But there are some that are more and more specific, like just cellular biology, just neuroscience, just plant botany, plant interactions. So yeah, there are all kinds of different journals out there. So that's one way. Another way to communicate science, again, is like seminars. You go to conferences or symposiums, and sometimes people will present to like a big audience, or there will be poster sessions, and it really is kind of like a science fair. It's really cool. You get to see what all kinds of other people are researching in this field, and get to know something a little bit more quickly, I guess. So it is really important to communicate with each other, and scientists do check each other's claims by performing similar experiments. If experimental results are not repeatable, like nobody else can get the same result, the original claim usually has to be revised. It's like scrutinized and like, hey, why can't anybody else get the same results as this one group? Did they fudge their data? Did they kind of mess with stuff and they're presenting something that's actually not really true? And that's a huge no-no in science. You can like lose your whole career. People have changed career fields entirely because nobody else will like trust them or respect them, basically, if they are falsifying data. So don't ever do that. So yeah, if experimental results are not repeatable, then it's very, suspicious. The original claim will have to be revised. Yeah, like I'm saying, scientists hold each other to a very high standard. It's not unusual for different scientists to work on the same research question, and then so, they'll know, like, hey, this is suspicious, this isn't adding up. Yeah. And so in doing science, scientists will usually study model organisms. So we can't always study stuff directly in humans, even if it's something we're super interested in. Human subjects are it's really hard to do anything in human subjects. You have to have, ethical methods and reasons, and there's some ways that it's not possible to do something that's ethical in a human on what you're studying. So for example, in neuroscience and studying the brain, so we'll use other organisms that we call model organisms to help like study something in a different kind of organism. And by learning from that organism, we can kind of make extensions to other things. So for example, the fruit fly that, annoys us all is actually a big model organism, not literally, but a popular one, especially in genetics. So studying the genetics of like their eye color and how that gets passed on. Is that dominant or recessive? The wings, all kinds of stuff. Another example is Caenorhabditis elegans. So it's this tiny, tiny microscopic roundworm. This is, a much zoomed in picture. And this is what I worked with in my master's program. So that was my model organism. So I studied the neurobiology of these guys because they have dopamine receptors that are really close in like genetics and structure to our dopamine receptors. And so if I can study them in these worms, then that'll help teach me a little bit more about what it might be similar to in humans. And so I was able to do research on gene editing on these tiny little worms and introduce a gene into their genome, and they would glow where certain dopamine receptors were expressed. And so nobody's going to really have a problem with that, right? They're just tiny worms. So that's why we use model organisms, because they're easier to work with. They usually grow fast. They're, tiny, easy to work with. And then there's also many other groups that are studying this organism. So there's a lot known about this kind of organism. Like, for example, in the worm, we know every single neuron by name. There's 302 neurons. And because so many groups have studied it, we know every single one by name. And so there's a lot of tools out there by being able to have various groups study the same model organism. There's a lot of resources that you can each draw on. So yeah, sometimes biologists approach interesting questions from many different angles. And there's a huge value to all this communication with scientists around the world. Many important inventions have occurred where a mix of different cultures ignited new ideas. So for example, the printing press was in part on innovations from China, so like paper and ink. And a combination of that and from Europe, the mass production mills, these ideas came together to make this new innovation. So having diverse backgrounds and viewpoints is really, really valuable in having this communication between scientists. So there were scientists all over the world also studying C. elegans from, all over the world, every continent basically. So the more voices that are heard in science, the more robust, valuable, and productive the scientific interchange. Okay, so that is the end of your first lecture. This is part 2. So hopefully after this lecture, you can list the major characteristics of living things, summarize how evolution accounts for the unity and diversity of life, and discuss the scientific process.